Hello and welcome, dear students. Today we are going to discuss about the topic genetic markers, utility of genetic markers in forensic science. The main objectives of today's lecture are to know the basics of genetic markers, to know the types of genetic markers, to know about single nucleotide polymorphism, that is SNP, as genetic markers, to understand the utility of genetic markers in forensics, to know about the role of restriction fragment length polymorphism, that is RFLP, in forensics, to understand the role of SNPs in forensics. Dear students, let's start with the introduction first. A genetic marker is a DNA sequence with a known physical location on a chromosome that can be used to identify individuals or species. It can be described as a variation which may arise due to mutation or alteration in genomic loci that can be observed. A genetic marker may be a short DNA sequence, such as a sequence surrounding a single base pair change, that is single nucleotide polymorphism, or SNP, or a long one like mini satellites. Genetic markers can help link an inherited disease with the responsible gene. DNA segments close to each other on a chromosome tend to be inherited together. Genetic markers are used to track the inheritance of a nearby gene that has not yet been identified, but whose approximate location is known. The genetic marker itself may be a part of a gene or may have no known function. Genetic markers consist primarily of polymorphisms, which are discontinuous genetic variations that divide individuals of a population into distinct forms. For example, AB versus ABO blood type, or blonde hair versus red hair. Genetic markers play a key role in genetic mapping, specifically in identifying the position of different alleles that are located close to one another on the same chromosome and tend to be inherited together. Such linkage groups can be used to identify unknown genes that influence disease risk. Technological advances, especially in DNA sequencing, have greatly increased the catalog of variable sites in the human genome. Multiple types of polymorphisms serve as genetic markers, including single nucleotide polymorphisms, simple sequence length polymorphisms, and restriction fragment length polymorphisms. In the human genome, the most common types of markers are SNPs, STRs, and indels. SNPs affect only one of the basic building blocks, that's adenine, guanine, thymine, or cytosine in a DNA segment. For example, at a genomic location with the sequence ACC-TGA, in most individuals, some persons may contain ACG-TGA instead. The third position in this example would be considered an SNP, since there is a possibility of either a C or a G allele occurring in the variable position. Because every individual inherits one copy of DNA from each parent, every person has two complementary copies of DNA. As a result, in the example, three genotypes are possible: homozygous, that is capital C, two copies of the capital C allele at the variable position; heterozygous, that is CT, 1C and 1T allele; and homozygous, that is TT, 2T alleles. The three genotype groups can be used as exposure categories to assess associations with an outcome of interest in a genetic epidemiology setting. STRs are markers in which a piece of sequence is repeated several times in a row, and the number of repeats is variable within and across individuals. Dear students, let's discuss about the background of genetic markers. For many years, gene mapping was limited to identifying organisms by traditional phenotype markers. This included genes that encoded easily observable characteristics or traits such as blood types. The insufficient number of these types of characteristics or traits in several organisms limited the mapping efforts that could be done. This prompted the development of gene markers which could identify genetic characteristics that are not readily observable in organisms, such as protein variation. Dear students, let's discuss about the types of genetic markers. Molecular genetic markers can be divided into two classes. Biochemical markers. The biochemical markers detect the change or variation at the gene level, such as amino acids. Biochemical markers can further be classified as allozymes and isozymes. Allozymes can be defined as an allelic form of an enzyme which can be distinguished by electrophoresis. Although it is an enzyme that are coded by different alleles at the same locus, isozymes can be identified as an enzyme that appear in two or more structural form, but these can be identified and separated from each other by their activities. Number two is the molecular markers. Molecular markers detect variation at the DNA level, such as nucleotide change, that is deletion, duplication, inversion, or insertion. Markers can exhibit two modes of inheritance, that's dominant, recessive, or codominant. If the genetic pattern of homozygotes can be distinguished from that of heterozygotes, then a marker is said to be codominant. Generally, codominant markers are more informative than the dominant markers. Dear students, let's discuss about the mitochondrial DNA, that is mtDNA, as genetic marker. Mitochondrial DNA is an extrachromosomal genome in the cell mitochondria that resides outside of the nucleus and is inherited from mother with no paternal contribution. Due to higher evolutionary rates of mitochondrial DNA relative to the nuclear genome, this marker is preferred in constructing phylogenies and inferring evolutionary history, and is therefore ideal for within and between species comparisons. The drawbacks of mitochondrial DNA analysis include hybridization, introgression, and incomplete lineage sorting. Moreover, mitochondrial DNA is of little use in investigating the recent loss of genetic variation and any individual level events such as identity, individual dispersal, and matting systems. Dear students, let's know about the restriction fragment length polymorphisms, or RFLPs. The RFLP is a technique that is not widely used now, but it was one of the first techniques used for DNA analysis in forensic science and several other fields. The RFLP is defined by the existence of alternative alleles associated with restriction fragments that differ in size from each other. The molecular basis of the RFLP is that nucleotide-based substitutions, insertions, deletions, duplications, and inversions within the whole genome can remove or create new restriction sites. Despite the fact that it is less widely used now, there have been numerous benefits to RFLP analysis. It plays an important role in allowing scientists to map the human genome as well as provide information on genetic diseases. RFLP analysis is useful to find out where a specific gene for a disease lies on a chromosome. This is performed by looking at the DNA from a set of family members who suffer from a certain disease and then searching for RFLP alleles that share the same type of inheritance pattern for the condition. Using RFLP analysis enables scientists to determine others who might be at risk for the disease or a carrier of the mutated gene. The RFLP is also one of the first methods used for genetic typing, also known as genetic fingerprinting, profiling, or testing. Despite that, the RFLP have many benefits, but it is still a slow and more tedious process compared to some of the newer DNA analysis techniques. It also requires substantially larger sample sizes than other forms of analysis. Dear students, let's now discuss about the random amplification of polymorphic DNA, or that's rapid RAPD. In the last decade, the RAPD technique based on the polymerase chain reaction has been one of the most commonly used molecular techniques to develop DNA markers. The RAPD technology provides a quick and efficient screen for DNA sequences based polymorphism at a very large number of loci. The major advantage of RAPD includes that it does not require pre-sequencing of DNA. The principle of RAPD is that a single short oligonucleotide primer which binds to many different loci is used to amplify random sequences from a complex DNA template. This means that the amplified fragment generated by PCR depends on the length and size of both the primer and the target genome. Since the advantages of RAPDs are the technical simplicity and the independence of any prior DNA sequence information, it is viewed as having several advantages compared to RFLP and fingerprint. A disadvantage of RAPD markers is the fact that the polymorphisms are detected only as the presence or absence of a band of a certain molecular weight with no information on heterozygosity besides being dominantly inherited, and also show some problems with reproducibility of data. Dear students, let's talk about the amplified fragment length polymorphism or AFLP. AFLP markers have found the widest application in analysis of genetic variation below the species level, particularly in investigations of population structure and differentiation. AFLP methods rapidly generate hundreds of highly replicable markers from DNA. Thus, they allow high-resolution genotyping of fingerprinting quality. The time and cost efficiency, reproducibility, and resolution of AFLPs are superior or equal to those of other markers like RAPD, RFLP, and microsatellites. AFLP markers have emerged as a major new type of genetic markers with broad application in systematic pathotyping, population genetics, DNA fingerprinting, and quantitative trait loci mapping. However, AFLPs are dominant biallelic markers and are unable to distinguish dominant homozygous from dominant heterozygous individuals. The AFLP method is an ideal molecular approach for population genetics and genome typing. It is consequently widely applied to detect genetic polymorphisms, evaluate and characterize genetic resources. Dear students, let's now discuss about the microsatellites. Microsatellites are simple sequence repeats or SSR loci, which have been referred to in the literature as variable number of tandem repeats or VNTRs and simple sequence length polymorphisms or SSLPs. These are found throughout the nuclear genomes of most eukaryotic and to a lesser extent in prokaryotes. Microsatellites range from 1 to 6 nucleotides in length and are classified as mono, di, tri, tetra, penta, and hexanucleotide repeats. The sequence of di, tri, and tetranucleotide repeats are the most common choices for molecular genetic studies. They are tandemly repeated, usually 5 to 20 times in the genome, with a minimum repeat length of 12 base pairs. The number of repeats is variable in population of DNA and within the alleles of an individual. In linkage studies, one can examine large number of families and see when the alleles of specific markers are inherited together with a phenotype in more cases than not. This approach assumes that a certain quantitative trait or character was affected by many unknown genes. So this approach is looking for associations between the variation of alleles and quantitative trait at the neutral DNA markers. The DNA marker is located on a chromosome and its inheritance can be monitored. Microsatellite markers have several advantages as they are considered to be robust and more variable and informative than RFLPs, RAPD, and AFLPs. Using the technique of polymerase chain reaction or PCR based require only low quantities of template DNA. Moreover, these markers are considered to be the best marker system for the detection of intervarietal polymorphisms. They are also useful for parentage analysis and for estimating the degree of relatedness of individuals or groups. On the other hand, these markers have several disadvantages as they are expensive, laborious, and time-consuming. Dear students, let's talk about the single nucleotide polymorphisms now, or SNPs. In 1996, Lander proposed a new molecular marker technology named single nucleotide polymorphism, or SNP. When a single nucleotide like adenine, thymine, cytosine, or guanine in the genome sequence is altered or changed, this will represent the SNP. In other words, it refers to a sequence polymorphism caused by a single nucleotide mutation at a specific locus in the DNA sequence. This sort of polymorphism includes single base transitions, transversions, insertions, and deletions, and the least frequent alleles should have a frequency of 1% or greater. Currently, SNP markers are one of the popular approach, despite they can be considered as a step backwards. Simple biallelic codominant markers when compared to the highly informative multi-allelic microsatellites. The more recent SNP concept has basically arisen from the recent need for very high densities of genetic markers for the studies of multifactorial diseases. The fundamental principle of SNPs is to hybridize detected DNA fragments with high-density DNA probe arrays, also called SNP chips. The SNP allele is then named according to the hybridization results. The SNP are third-generation molecular marker technology coming after RFLPs and SSRs. On the other hand, the most important disadvantage of single nucleotide polymorphism, or SNPs, is the low-level information obtained as compared with that of a highly polymorphic microsatellite, but this can be solved by using a high number of markers, that is SNP chips, and whole genome sequencing. Variations in the DNA sequences of humans can affect how humans develop diseases, respond to pathogens, drugs, etc. However, the greatest importance in biomedical research is for comparing regions of the genome between cohorts, such as with matched cohorts with and without disease. Dear students, let's know about the DNA barcoding markers. A DNA barcode is a short DNA sequence from a standardized region of the genome used for identifying species. The essential aim of DNA barcoding is to use a large-scale screening of one or more reference genes in order to assign unknown individuals to species and to enhance discovery of new species. Biological taxonomists apply this principle to species classification. The first application of using the DNA sequences in systematical biological taxonomy, also called DNA taxonomy, was conducted by Tots, and then Herbert proposed the concept of DNA barcoding. This approach has some disadvantages represented by the genome fragments are very difficult to obtain and are relatively conservative and have no enough variations. Dear students, let's understand the uses of genetic markers. Genetic markers can be used to study the relationship between an inherited disease and its genetic cause. For example, a particular mutation of a gene that results in a defective protein. It is known that species of DNA that lie near each other on a chromosome tend to be inherited together. This property enables the use of a marker which can then be used to determine the precise inheritance pattern of the gene that has not yet been exactly localized. Genetic markers are employed in genealogical DNA testing for genetic genealogy to determine genetic distance between individuals or populations. Uniparental markers or mitochondrial or Y chromosome DNA are studied for assessing maternal or paternal lineage. Autosomal markers are used for all ancestry. Genetic markers have to be easily identifiable, associated with a specific locus, and highly polymorphic because homozygotes do not provide any information. Detection of the marker can be direct by RNA sequencing or indirect using allozymes. Furthermore, molecular genetic markers were used to resolve the issue of natural transmission, the breed of origin, that is the phylogenetics, and the age of the canine tumor. Dear students, let's now discuss about the utility of genetic markers in forensics. Like other subjects, forensic genetics gradually emerged and developed through long-term social practice. Following the discovery of the ABO blood groups by Carl Landsteiner in 1900, human blood type was used in identification, and forensic genetics entered the scientific age. In 1910, the French criminologist Edmond Locard proposed the Locard's exchange principle and stated that every contact leaves a trace, which laid the foundation for modern forensic sciences. In 1926, Thomas Hunt Morgan proposed a theory of genes which provided the basis for the development of forensic genetics. In 1953, the discovery of the double helical structure of DNA by Watson and Crick enabled the start of forensic genetics research at the molecular level. One of the central aspects of forensic genetics is the use of genetic markers, which are the easily identifiable phenotypes of genotypes. Genetic markers generally have features such as strong polymorphism, codominant expression, and ease of observation and recording. With the development of genetics, the use of genetic markers has also developed steadily. To date, the development of genetic markers has gone through 4 major stages characterized by the use of morphological markers, cytological markers, biochemical markers, and molecular markers. During the early 1990s, short tandem repeats or STRs were used for the first time as the key molecular markers in human paternity testing. STR typing based on capillary electrophoresis can only reflect differences in length. This provides a new possible method for difficult cases involving complex kinship identification and the resolution of mixtures. Since forensic polymorphic STR loci are limited in number and cannot provide more functional information, this hampers the effectiveness of the application in forensic community. Dear students, let's now talk about the role of SNPs in forensics. A new type of genetic marker, single nucleotide polymorphism or SNPs, may replace STRs in the future, although this process may take a considerable period of time. This amplification product of individual SNPs sites can be very short, which makes SNPs suitable for the analysis of highly degraded forensic samples. SNP analysis already plays an important role in the identification of individuals after mass disasters. Furthermore, SNPs are linked with multiple phenotypes such as individual skin color, scleria color, hair curliness, information concerning ethnic origin, differences in alcohol metabolism, and susceptibility to hereditary diseases. These advantages of SNPs have all been successfully applied, and SNP analysis occupies an unsurpassed position in forensic practice. SNPs have historically been used to match a forensic DNA sample to a suspect, but has been made obsolete due to advancing STR-based DNA fingerprinting techniques. However, the development of next-generation sequencing or NGS technology may allow for more opportunities for the use of SNPs in phenotypic clues such as ethnicity, hair color, and eye color with a good probability of a match. This can additionally be applied to increase the accuracy of facial reconstructions by providing information that may otherwise be unknown, and this information can be used to help identify suspects even without an STR DNA profile match. Some cons to using SNPs versus STRs is that SNPs yield less information than STRs, and therefore more SNPs are needed for analysis before a profile of a suspect is able to be created. Additionally, SNPs heavily rely on the presence of a database for comparative analysis of samples. However, in instances with degraded or small volume samples, SNP techniques are an excellent alternative to STR methods. SNPs, as opposed to STRs, have an abundance of potential marker, can be fully automated, and a possible reduction of required fragment length to less than 100 base pairs. Dear students, let's now talk about the RNA level genetic markers. Apart from DNA level genetic markers, RNA level genetic markers have also become a new type of marker and research hotspot in forensic genetics. In 2000, progress in RNA research was acclaimed by science as a major scientific breakthrough. A series of groundbreaking advances in RNA research during the past 20 years have enabled RNA to escape from the brilliance of DNA's aura. This has changed RNA from the supporting role to the leading role and has made RNA a new challenge to DNA's central position. In the field of forensics, steady progress is being made in applied research involving the use of RNA to identify racial affiliation, the identification of bodily fluids, the determination of time of death, and the analysis of injuries. In addition, having the advantage of convenience, economy, speed, and precision, the protein-level genetic markers offer great promise in forensic genetics and related fields. Dear students, let's now conclude the lecture. The application of genetic markers has advances in pace with the scientific knowledge and technological capabilities, which has expanded and changed the scope of forensic genetics research. From the dual perspectives of the development of genetic markers and their applications, it is evident that forensic genetic research is no longer limited to DNA level markers, but also includes RNA level and protein level markers. The scope of forensic genetics research is no longer limited to conventional determination of kinship and individual identification. Instead, it has steadily branched out into areas such as forensic clinical medicine, forensic pathology, and forensic psychiatry, yielding many new findings and applications. In forensic science, the genetic markers are used for the detection of short tandem repeats, and restriction fragment length polymorphisms are used in DNA profiling of an individual. They are used to solve paternity disputes and are useful in gene mapping. Also, these help in the characterization of genetic diversity. Dear students, it was all about today's lecture regarding genetic markers, utility of genetic markers in forensic science. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.